The human T lymphotrophic virus 1 was the first human pathogenic retrovirus discovered, and it causes adult T-cell lymphoma as well as HTLV-1 associated myelopathy. The retrovirus is a type of RNA virus like human immunodeficiency virus. The structure of a retrovirus consists of an envelope composed of lipids, a dimer RNA, and proteins. HTLV types 1 and 2 are presumed to have derived from primate T lymphotrophic viruses. The development and clinical impact of human T lymphotrophic virus 1 remains unknown, and the virus affects about 5 to 20 million people worldwide, mainly in southern Japan, the Caribbean, South America, Central and South Africa, the Middle East, and Central Australia. The average age of people with the virus is 40 to 50 years old, and there is a higher frequency of women than in men. Many individuals affected with human T lymphotrophic virus 1 remain asymptomatic. In a 2004 study, human T lymphotrophic virus 1 patients had a higher frequency of bladder or kidney infections and arthritis. These patients also reported several opportunistic co-infections and had increased numbers of neurological symptoms. In a similar manner to HIV, HTLV-1 can be transmitted through sexual contact, intravenous drug use, or sharing contaminated needles, blood transfusions, and breastfeeding from mother to child. It cannot be transmitted through casual contact, such as shaking hands, hugging, or sharing restrooms. Sexual transmission of HTLV-1 appears to be more efficient from males to females. The human T lymphotrophic virus 1 is present around the world and causes many different diseases. Pulmonary disease have been identified in various asymptomatic HTLV-1 patients in Japan in a 2006 study. While Japan has a large percent of patients with human T lymphotrophic virus 1, Australia is another region of interest. HTLV-1 infections in Australia have been observed in the early 1990s, but a high number of infection rates in Central Australia have not been known about until recently. While actual prevalence rates are unknown, there are estimates ranging from 14 to 60% in some indigenous communities. A 2011 study reported a link between the human T lymphotrophic virus infection and pulmonary disease in Central Australia. Evidence of pulmonary disorders have been reported in 50% of patients with HTLV-1 associated myelopathy and in 30% of asymptomatic patients. The reason for such a high prevalence of pulmonary disease remains very unclear. Very little research has been done to eradicate human T lymphotrophic virus 1, and knowledge on the global commonness of HTLV-1 is inadequate. The risk of the virus to the public health worldwide has been misjudged, and very little research has been done to control the outbreak in Central Australia. There is no likelihood of vaccines for this disease, and the screening of blood banks is not universal. HTLV-1 virus is a lifelong disease, and many infected individuals remain asymptomatic throughout their lives. The HTLV-1 infection is usually detected by screening for antibodies against the virus in the blood or cerebral spinal fluid. There is no specific treatment to date. Reverse transcriptase inhibitors, which are a class of antiretroviral drugs used against human immunodeficiency virus 1 and inhibit the activity of viral DNA polymerase that is required for replication, were not effective against HTLV-1 in clinical trials. Without a vaccine available, the virus has been hard to contain. Patients found with the disease should refrain from donating blood, organs, and other tissues. They should also refrain from sharing needles or syringes. Screening by blood donations have been found to be an effective strategy to prevent the human T lymphotrophic virus 1. Pregnant women should be screened for the virus and avoid breastfeeding to reduce maternal transmission if they are found positive. References on how to prevent sexually transmitted infections should be indicated, including condom use and avoiding multiple or unfamiliar sexual partners.